Hey guys, welcome to the video about the stimulus response model. All right, so let's have a look at the points from the study design, then we can get into the detail. So we need to know what the stimulus response model is in terms of signal transduction and a three-step process. Okay, so we're going to learn a three-step process. It's rare that you get asked questions just on the stimulus response model. Usually it's part of the second dot point that I have here. So that's why I've got both. So the difference in signal transduction for hydrophilic and hydrophobic molecules or signals in terms of where they come and where they get in there. So this video is just going to be a brief thing on the stimulus response model. And then the next video is the one that you get asked all the questions on. So we'll go through this and then we'll pause and we'll go through the next video. So what do we mean by stimulus response? So if you think all the way back to year 9, you probably saw this kind of pathway that talked about effectors and responses and stimulus and receptors and all of those bits and pieces. Now, we don't need to know that the way that we taught it in year 9, it's a little bit different. We're just breaking it down into 3 main sections. So the first section that we're looking at here is reception. The next section is transduction. So that's when you've got your signal and it's been received and you've got to pass it on. And then the final part that we look at is cellular response. So they're the 3 stations that we look at. So in your notes on page 3, there is a diagram and it says to annotate the diagram as we go through. Now there is a table underneath that enables you to fill it in. You can go through that. If you want to draw anything else on your diagram, draw anything else as we go through. So the first thing is all about reception. Lo and behold, here is another scenario where you have a molecule that fits into another molecule perfectly. Okay, so reception is where your signaling molecule binds to a receptor. Okay, that means your signaling molecule has a particular shape. That means your receptor has a, you guessed it, complementary shape. Okay, so we've got this complementary shape here. So that is all reception is. It's just the binding of the signal to the receptor. Okay, let's go through that. So here we go, the detection of a signaling molecule by a cell. Now, the difference is where those receptors can be located. Now, they can be located on the outside of the plasma membrane. So what I would like to change this to say is I would like to change this to say the outside of the plasma membrane, okay, or the extracellular surface. You can also say that too. Extracellular surface. Now, the reason that's important is because this molecule, you need to really let the examiners or anyone that you're telling know that it's not going to get through that plasma membrane if it's binding on the plasma membrane. So if you say on the plasma membrane, are you sure? So if this is your plasma membrane, are you sure if it's going to be binding on the outside here or the inside here? You need to be clear in the way that you write it. All right. The next place that the receptors could be located is in the cytosol, and I'm coming back to all of these in the second video that we're going to do. And the third place is in the nucleus itself. So the position of that molecule is completely dependent on whether or not it is hydrophilic and hydrophobic. And I reckon already you're going through your brain, you're going, okay, I reckon I know which one's which. Which one's going to bind in the cell and which one's going to bind outside the cell. When you're talking about receptors, we use this brilliant keyword that we've never seen before, and that is the fact that receptors are specific. Yes, and they only bind to particular signaling molecules. So the receptor, this bit here, is complementary in shape to the signaling molecule. And here you have the nice little red signaling molecule, this one here, that is your stimulus. That is your molecule that's going to bind to that receptor. The next thing that happens is the process of transduction. Now, in one of the 2 pathways you're going to learn, transduction's really easy. The other pathway, you might find it a bit weird. But transduction is all about passing messages. So it's all about relaying the signal or passing the message on. Okay, when it reaches its final destination, that's when it's going to bring about that cellular response. And this is where we come into this part here. We talk about these cascades, and I'm going to bring back cascades when we talk about apoptosis, and I'll bring them back when we talk about it in the next video as well. So the last stage of signal transduction, or this stimulus response model, is of course the response. So you've had your stimulus, it's bound to your receptor, you've gone through transduction and moved that signal through your cell, and then something is going to happen. So this is when something happens. Now, what could that something be? That something could be transcription. So it could initiate transcription. It could initiate activating enzymes. It could initiate secretion of proteins. So for example, a hormone binds to a particular receptor on a cell, it goes through transduction, and the cellular response is that that cell now produces another hormone. So it's all about going through that process for a bunch of different examples. And that's why you're rarely asked about this process in itself. You might be asked to identify whether or not it's reception, transduction, or a response from an example that they give you, but you would never really be asked to talk about the processes in general, because they don't make sense unless you have an example. So what we've done right now is we've gone through this dot point here, which is the stimulus response model when applied to the cell in terms of signal transduction. We've got 3 steps: reception, transduction, and cellular response.